In this video, we will discuss about some of the in vivo results where HFD can enhance highly metastatic tumor cells to metastasize to lungs. So what they did is they inject the tumors at the mice, at the tail of the mice, and then they see the metastasize of these highly metastatic tumors. So initially, when they check the in vivo results of the tumor growth in the high-fat diet mice and in the nude mice, that is control mice, they are able to see that tumor growth is significantly higher in the case where high fat diet uh, mice are being injected with these metastatic tumors. And thereafter, they also check this tumor spreadness using p53 staining, and they are able to see that in the case of high fat diet, the staining is more and is significantly higher compared to the control case. And this thing, when they quantified in the metastatic foci per lungs, they are able to see that the high fat diet, the metastasis tumors are higher compared to the control case. And one more interesting thing that they are able to find out is that most of these tumors are in the form of single cells or double cells compared to the case where group of cells are migrated together. And then in order to avoid the effect of different initial size of tumors, what they did is they took the almost similar size of tumors and check almost similar size of tumors. They have injected almost similar size of tumors in the control mice and in the high fat diet mice, and then check the metastatic foci per lungs, and they are able to, in the immunostaining image and in the penalty, we can see that the size of the tumors in the HFDs are higher compared to the control case. And when they check the metastatic signals in the lungs, they are able to see that tumors metastatic tumor signals are higher in the HFD mice conditions compared to the control mice conditions. And these things are marked by the black arrows, which are higher in the HFD conditions compared to the control conditions. And when they also check these metastatic foci per lung, they are able to see the similar results, that is, cells metastasized into the lungs are higher in the HFD conditions compared to the control conditions, and most of these metastatic cells are in the either single or doublet. And now they wanted to check the early effect of this metastatic. So for this, what they did is they used these C57BL6 mice and then uh, feed one mice with high fat diet, it is 60% kilocalorie fat, and another one with is a control, and then they inject the mice in the tail of the then they inject the tumor in the tail of the mice and sacrifice the mice after 1 hour. So for the diet, they treated the mice with the high-fat diet and the control conditions for 12 weeks. After that, they inject these tumors at the tail of the mice, and after 1 hour, they sacrifice the mice. This is the actual scenario. And then they check and in this case, they inject the tumors with GFP luciferase. And when they check that signals, they are able to see that in the control case, the signals are not that much spread across different organs, but in the case of HFD case, the signals are spread across almost most of the lungs area. And these things they have quantified using this in the panel I, where we can see that the signals for in the HFD conditions are significantly higher compared to the control conditions. And when they did the similar experiment with highly metastatic tumor models, they are able to see is similar results, that is single cell mice, that is single cell metastasis is higher in the HFD conditions. Thereafter, the small metastasis and the micrometastasis compared to its corresponding control conditions. And when they check the overall metastatic foci per lungs, they are able to see that the number of cells spread in the lungs are higher in the HFD conditions, and that too they are spread in the form of single cells or doublets compared to a group of clusters. And when they check the similar studies, when they did the similar studies with low metastatic mice models, they do not see any too much significant. They do not see the significant difference between control and the HFD diet conditions mice, indicating that it's the metastatic size also plays an important role along with the high fat diet for the metastasis of the tumor cells, and those results are shown in the supplemental figures. So I hope you understood how they come up to this point that HFD enhances highly metastatic tumor cells in the to metastasize to lungs. If you need to understand more about these experiments in detail, you can go through the reference that has been described in the descriptions. That is mentioned in the descriptions. And they also did so they did the experiment mainly on the breast cancer cells that metastasize to the lungs. So this is one of the point that you need to aware of what kind of tumor cells they used. So thank you for your time.